All right, good afternoon, everybody, or good evening if you're in Italy. It's a pleasure for us to welcome our seminar speaker for today, Dr. Fabrizio di Fagagna. Sorry about that. So Fabrizio is actually performed his Bachelor's of Science at the University of Trieste in Italy, and then his PhD at the same institution, and then went on to the International School for Advanced Studies in Trieste, and then the Gurdon Institute in Cambridge University for his postdoctoral studies. And then started his own group as a principal investigator at the FIRC Institute of Oncology, or the IFOM, in Milan, and is also a group leader at the National Research Council in Pavia in Italy. So it's wonderful to have Fabrizio here with us today. And he's going to tell us about non-coding RNAs and DNA damage silencing. So welcome, Fabrizio.
Thank you.
Want to share your screen?
I'll share my screen. Do tell me if you see full screen my slide, the first slide.
Yes, that looks good, yeah.
Fantastic. So thank again, Frank and Jay and everybody that made this possible for me. It's an honor, it's a pleasure to join the non-coding RNA community. We are sort of an outcast group, meaning we are historically a DNA damage lab. Then we had to become an RNA lab, and in a second you'll see why. So as I said, we have been a DNA damage lab for many years, and we study specifically what happens in a cell, mammalian cells, when DNA damage is generated, and everything that happens in a cell is known as the DNA damage response, which is essentially, it's a signal transduction cascade, which stems from the initial recognition of a lesion by a complex known as the MR11, RAD50, MBS1. So this protein senses DNA breaks, and then it recruits a large signaling molecule, a protein kinase, ATM, which is then brought to the sites of DNA damage, starts to phosphorylate a substrate. The first substrate is a chromatin associated protein. It's a histone variant. It's called H2AX. When it is phosphorylated, we call it gamma H2AX. And then gamma H2AX acts as a beacon, as a Velcro that will establish a positive feedback loop, because then gamma H2AX will be recognized by mRNA again. So mRNA now binds a gamma H2AX, which recruits more ATM, which phosphorylate more gamma H2AX. So eventually you spread this chromatin phosphorylation event for 100 of kilobases. This is a very robust signaling event. And in fact, if you damage cells, for instance, by exposing them with to ionizing radiations, you see these dots, we call them foci. Each one of them is an individual DNA lesion, an individual DNA double-strand break, which has been labeled by this chromatin modification event. And here DNA damage signaling starts, of course, with the aim of repairing such lesion. In addition to gamma H2AX and ATM, there are a bunch of proteins which accumulate to sites of DNA damage. 53BB1 is one of them, and you will be hearing me talking about it. And these are local events, but of course, eventually the signaling needs to spread throughout the nucleus. So this is the job, instead of more diffusible kinases such as CHEK2 or CHEK1. I did not mention, but there is a parallel signaling pathway which stems not from the recognition of double-strand breaks, but rather by patches of single-stranded DNA. So eventually you get this further amplification, this diffusible kinases further phosphorylate additional substrate. Eventually somebody must take decisions. P53 is one of the strongest decision makers, and cells either decide to kill themselves, we call it apoptosis, or they transiently arrest or permanently arrest. So we have been working on this for many years, and then we realized that in fact pretty much everything that I described you would not take place if you do not allow the synthesis and the processing of non-coding RNA, which are generated at the sites of DNA damage. Things are complicated. Certainly they were complicated for us because we were not an RNA lab. So I thought, just to use it in the best possible way, the time that I was generously allocated today is to synthesize our understanding of these events in an animated cartoon, which is the next slide. I will then show you some key experiments, key results that support this model. So this will allow me to show you some of our most recent results, and including some unpublished data. So what we think is going on is that when DNA damage is generated, This is initially recognized by the sensor complex that I mentioned before, mRNA. So far, so good. That is very canonical. However, then we discovered that mRNA loads RNA-POL2 at the sites of DNA damage. This is an active RNA-POL2 complex that will synthesize what we called damage-induced long non-coding RNA, both from the break and going toward the breaks. These long non-coding RNA are then further processed by the same machinery that processes microRNA, meaning DROSHA and DICER. And these shorter RNA, we call them ddRNA. And then we demonstrated that this ddRNA can bind back to the unprocessed complementary single-stranded D-link RNA, and only when the small RNA are bound to the long complementary D-link RNA, this allows the formation of the DNA damage response foci as we know them. 53BB1 is one of the components that I've highlighted here. So this is the way we see this event. Now let me show you some of the evidence again in support of this. Number one, is there actual transcription going on at the sites of the damage? Is it triggered? So here is the evidence both at endogenous sites and at sites that we have engineered. So we generated double-strand breaks in many different ways by using this meganuclease or this restriction enzyme or even CRISPR-Cas9, and both in genic and intergenic regions, you see there is an induction of transcription. This is the ratio of the transcription before damage and after damage. So there is an increase of transcription on both strands, and this happens also at these sites, again, which are exogenous integrated sites. We thought they were useful to be tested because in these cases, obviously, they are not associated with any transcription or promoter activity. Usually, they are bacterial plasmids which are being integrated. So transcription can be detected upon DNA damage generation by RT-qPCR, but also by single molecule FISH, and this is the result of very fruitful collaboration that we had with the Setu Pichaya and in the lab of Niels Water. And as you can see here, with the set of probes spanning upstream and downstream of a double-strand break that we generated again, one of these artificial loci, you can see that in the uncut situation, then upon generation of DNA damage, you see a very clear increase of transcription. So by two different means, we can detect DNA damage-induced transcription at sites of DNA damage, but more conveniently, we could reproduce these observations in vitro, meaning that we used a plasmid, and this is a very dumb plasmid that doesn't contain any transcriptional promoting activity. So this is PUC. And then we incubated this plasmid in a mammalian self-nuclear extract that we spiked with some hot UTP. And what we observed that this plasmid, when it is incubated with this nuclear extract in its circular form, as expected, doesn't really lead to any meaningful transcription. However, the very same plasmid, if it is linearized, now this leads to the induction of very clear transcript. These are real RNA products because they are DNase resistant, the RNase sensitive, and they are exquisitely sensitive to alpha-amanitin inhibition. So clearly these are RNA-POL2 products. We sequence these products and we map them around this plasmid, and indeed we observe that this transcript before linearization, when the plasmid is linear, you get the induction of both strands of the double-strand breaks of this transcript. We also map them by 5' rays, so they really start very close to the breaks. This system is very convenient, very robust, and so we further capitalize on the strength of this system. And very recently, we managed to reconstitute these events not using a whole cell extract, rather by using recombinant or purified components. So I show you that if we use this recombinant mRNA complex and purified RNA-POL2, and again, it's usually in the presence of a hot UTP, we observe that if we mix a linear DNA template and RNA-POL2, we do get some transcription, but nothing to be excited about. However, if we add also the mRNA complex, now transcription is very much, it's much more robust. This allowed us to further investigate how mRNA support transcription at DNA ends. So we used a bunch of mRNA mutants and drugs and inhibitors, and in a number of different ways, we came up with the demonstration that the way that mRNA promotes transcription at double-strand breaks is by melting DNA ends. In other words, mRNA loads RNA pool to at sites of DNA damage, then allows them to allow the separation of the two strands, and you end up generating something very, very similar to a transcription bubble, and therefore RNA pool 2 now is able to process and go away from the break. So the ability of mRNA supported transcription is mediated by its ability to melt DNA ends. So I explained you how mRNA we think it is working. I showed you the evidence of damage-induced transcription. I proposed you also that the link RNA are processed, and indeed we could show that both DROSHA and dicer are recruited at sites of DNA damage. This is our evidence. So this is a DROSHA, a ChIP-seq experiment in a cell line in which we can introduce double-strand breaks in different sites, and you see this nice peak of DROSHA accumulation upon DNA damage only. And luckily for us, we did not have to show that also dicer is recruited at sites of DNA damage, because somebody else showed that specifically the phosphorylated form of dicer is nuclear, and again, it is engaged at sites of DNA damage. So I showed you that the processing is taking place at sites of DNA damage, and the processed forms is what we call the ddRNA. We detect them by RT-PCR, but probably most convincingly, we managed to sequence them. That was a fairly complex effort that we managed only because we had the luck and privilege to work with Piero Carninci at the RIKEN in Japan that sequenced these products and demonstrated they are induced upon DNA damage, and they are reducing following the knockdown of dicer and DROSHA. What I also showed you is that this ddRNA, when they're processed, they bind back to the processed D-link RNA. So this is the interpretation that we proposed of the experiment that is shown in this slide, again, part of the collaboration with Setu and Nielsen. So what I'm showing you here is that we're using a cell line in which we can introduce a double-strand break in a region which is flanked by LAC repeats, so we can express LAC RFP, and so we see a green dot where we are about to generate a double-strand break. Now, if we microinject fluorescently labeled ddRNA, which have the sequence of these damaged sites, if the site is not yet damaged, this ddRNA are scattered throughout the nucleus. They do not accumulate there. Rather, if you induce a double-strand break, now you start to see the accumulation of this ddRNA at the sites of DNA damage. And this is not only DNA damage dependent, but it is also sequence specific, because other RNA with a different sequence will not accumulate there. And Setu also perform a stepwise photobleaching experiment to count how many ddRNA can accumulate at the sites of DNA damage. As you can see, this is the results before damage and upon DNA damage, and when you are using, in fact, ddRNA with a different sequence, they will not accumulate there. So ddRNA accumulate there at the sites of DNA damage. We think that they do so because they interact with a D-link RNA. And again, this is demonstrated, or this is the interpretation of this result, because this ddRNA will not localize at sites of DNA damage in the setup that I've just described you if you treat the cells with RNA pool 2 inhibitors. In other words, if you do not allow the synthesis of this D-link RNA, what we in the lab we call the platform, you would not allow the generation of this sort of landing platform where the ddRNA will pair, anneal, and therefore localize at sites. So this is, again, to recap what I told you: sites of DNA damage, the link RNA synthesis, processing, pairing between the short and the long RNA, and this leads to the formation of this foci. And in a second, I'll tell you how we think RNA contribute to the accumulation of these proteins at sites of DNA damage. However, I want to point your attention to the fact what I've just told you, that double-strand breaks are in fact very similar to bidirectional promoters, because they do trigger transcription on both sides of the break. So more recently, we started to wonder what is in fact the difference between a double-strand break and a canonical RNA-APOL2 transcriptional promoter. So we went back to the textbooks and we realized that, of course, the RNA-APOL2 doesn't travel alone. It is rather recruited, positioned, and oriented and activated by the pre-initiation complex, which probably you don't need to explain. It's made by many additional subcomplexes, starting from TF2D, which contain TBP, the tata binding protein, and TF2H, which is one of the last subcomplexes, which is recruited to promoter, containing CDK7 that will unleash RNA-APOL2 activity. Therefore, we tested, could it be that RNA-APOL2 is not only, well, that in addition to RNA-APOL2 to sites of DNA damage, also the pre-initiation complex is somehow engaged. So we performed the next experiment. We introduced a double-strand break by this enzyme, and then we perform a bunch of chromatin immunoprecipitation experiments with antibodies against the individual components of the pre-initiation complex, and then we quantified the amount of genomic DNA that we brought down in this chip experiment with primers spanning different distances from the break. And what we observed when we amplified DNA only 100 bases from the break, we observed that upon DNA damage generation, we get gamma-H2X as expected. We get RNA-APOL2, interestingly, not POL1, not RNA-APOL3, but we also got TBP and TF2A, B, E, F, H, mediator 1, CDK7, and CDK9. The entire pre-initiation complex is recruited to sites of DNA damage. Further away, 2 kb away, you just get the RNA-APOL2, you do not get the pre-initiation complex. And I remind you that D-link RNA are usually no longer than 2 kb. And in fact, 3 kb away from the break, you just get the gamma-H2X, you do not get RNA-APOL2, you do not get the pre-initiation complex. In other words, this really seems to me that when you generate a double-strand break, you assemble a transcriptional unit very, very similar to a canonical gene. You get a promoter, with its associated pre-initiation complex. You get the transcribe unit, and 3' of it, you don't get transcription anymore. Of course, this was happening at one site. We reproduced this at many other sites, but of course, we wanted to have a global view of these events just to make sure that we have not been lucky in making these observations. So we started a collaboration with Eli Rothenberg, which is extremely good in super resolution imaging of DNA damage response event. And so these are the results. So when he performed this analysis, he observed a very good correlation between gamma-H2X, our favorite marker of DNA damage, and the phosphorylated or activated form of RNA-APOL2. Importantly, our negative control is a very abundant DNA replication factor, MCM. So gamma-H2X co-localizes with the RNA-APOL2. The same applies to TBP and CDK7, a TF2H component. So the pre-initiation complex is globally recruited to sites of DNA damage. Is it relevant to such a recruitment? Indeed, what we decided to do was to knock down components of the pre-initiation complex and monitor the impact that it has on D-link RNA and on DDR foci formation. And this is the result. As soon as you knock down TBP or TF2B, while gamma-H2X is unaffected, this is something I should have mentioned before, all this RNA business that I'm describing to you is controlling events downstream of gamma-H2X. Another way of saying it is that foci are generated when you have gamma-H2X, the beacon, and RNA, which is sort of the glue which holds the protein together around the beacon. So you knock down TBP, gamma-H2X is unaffected, but 53B1 is completely lost. The same applies for phosphorylated, the reactivated form ATM. The same applies for MPC1. Of course, you are right in thinking that as soon as you knock down TBP, as soon as you knock out the pre-initiation complex, the entire expression of the cell is affected. So maybe many of these proteins are not even expressed. Of course, we check they are expressed, but of course you cannot rule out indirect effect. So what we needed to do was to inhibit the pre-initiation complex in an in vitro system and see whether this has an impact or not on DDR foci formation. Unfortunately, there were no systems in vitro that could recreate this foci formation, so we had to invent them. And conceptually simple, but it was not. So what we did was to generate some nucleosomes. So we wrapped DNA around histones, and we had either both ends of this DNA bound to streptavidin beads. So this is our intact, no double-strand breaks present DNA, or only one end is bound to the beads. The other end is the signaling end, this mimic a DNA double-strand break. And then we incubated these two set of nucleosomes into a nuclear extract. Then we did some pull down experiment, and we monitor what is recruited, what is associated to this exposed, damaged chromatin end. And of course, in this system, yes, we could inhibit pre-initiation complex component and see if this has any impact. And what we observed that, again, see, there are probably too many bands, but here, just trust me, what we could demonstrate that you recruit the same components that are recruited to sites of DNA damage, like MDC1, 50W1, and phosphate TM at the chromatinized DNA end, only when it is linear, only when you have H2IX, and when you allow the synthesis of RNA by fueling the synthesis with RNTP. This is very sensitive to an RNase treatment, so you lose the recruitment of these proteins at these DNA ends. But most importantly, if you now incubate this reaction with antibodies, which we demonstrated in vivo to be inhibitory for the functions of TBP or TF2B, now you impair the recruitment of these DDR factor sites of DNA damage. So this proves that the evidence that the results that we showed you in vivo, they were not indirect. Indeed, TBP makes a direct contribution, and we demonstrated through the synthesis of the ncRNA, to the recruitment of DDR proteins at sites of DNA damage. However, to go back to the initial question, what is the difference between a double-strand break and an RNA-APOL2 transcriptional promoter? We could not find any. Now, let me go to the question, because what I showed you before is that the RNA-APOL2 synthesizes this bunch of RNA transcripts that somehow fuel the accumulation of proteins at sites of DNA damage. How do they do that? Well, probably most of you are aware of the fact that RNA controls what now is being termed in many different ways, liquid-liquid phase separation, liquid mixing, or condensates formation. And indeed, just by looking at the behavior of this DDR foci, you can see that there is, these are different time points following DNA damage generation by ionizing radiation. You get a nucleation phase, then they grow in size, but not in number, and then they undergo coarsening. So again, very, very reminiscent to the behavior of liquid structure. So we started a collaboration with a group of physicists that helped us in characterizing the physical properties of this DDR foci. A simple thing was to take a movie of the behavior of this DDR foci. I'm going to show you, and these are 40 seconds in the life of DDR focus, so you can immediately appreciate how dynamic it is. So certainly this is not a static, solid, concrete, static again structure. We then decided to further characterize the behavior of this structure. We performed a FRAP experiment in order again to probe the internal dynamic and exchanges between phases of DDR foci. And again, upon FRAP experiment, within 10 seconds, as you can see here, you recover 50% of the signal. So a very fast recovery. And when we calculated, our collaborators calculated the diffusion coefficient of 50W1 within foci and outside foci in the nucleoplasm, it was, this diffusion coefficient was really 500 times greater than in the nucleoplasm. So our colleagues really, you probably are, you're probably aware that at this time, there are a number of concerns about the characterization of liquid features, and everything is liquid, and so strict quantitative measures are necessary. Again, we had the luck to collaborate with the physicists, which were extremely accurate, and certainly I will not be able to defend most of their results and the equations, but suffice to say that we could even manage to appreciate the capillary waves on the surface of the DDR foci. Again, once again, confirming the liquid nature of the structure, but probably the best way that I managed to convince them to run an experiment to prove whether the structures were solid or liquid was the following one. Actually, before telling you this experiment, I have to tell you that and this is going to be most of the main focus of the rest of my presentation, is that if you use an antisense oligonucleotide against the non-coding RNA that are generated at sites of DNA damage, you disrupt that individual focus. So with this tool, we perform the following experiment. So we microinjected these antisense oligos and we observed the dismantling of these DDR foci. And then we ask ourselves, how do they disappear, such foci? Do they disappear as solid or liquid structures? Because the physicists that we are interacting with explain me that if a solid is broken in pieces, you would get a loss of volume. However, the small bits will have the same density, meaning if you have a big boulder and you break it in pieces, the single pieces will still have the same density of stones. However, if it is if the solid that you are disrupting is liquid, you will have an initial drop of density and only then a loss of volume. Therefore, we treated our foci with antisense oligonucleotide, and then we measure what is happening. Are we losing the volume first, or are we losing the density first? And these are the results. Again, these are the quantitative measure. If you just focus on this, you observe there is an intensity decrease, which is much faster than the volume decrease. So indeed, these DDR foci are disrupted when you inhibit the function of this non-coding RNA with an antisense oligonucleotide, following the physical laws of a liquid, not of a solid object. So, we have added one small picture here. So now we know how DDR foci are regulated by RNA by generating this mesh of polyvalent interactions that hold proteins together. But of course, this also provides us an opportunity for, and this is the title of this slide, sequence-specific DDR inhibition. Because of course, you can see here, all these foci, they contain the same proteins. If you inhibit 53B1, you're going to inhibit 53B1 throughout the nucleus. However, what we are proposing is that each of these lesions has its own set of non-coding RNA generated locally in situ at the sites of DNA damage, and therefore an antisense against those non-coding RNA will be able to individually dismantle that focus without impacting signaling elsewhere in the nucleus. So we wanted to make this point, and so this is the experiment that hopefully will convince you. So we generated 3 double-strand breaks within the nucleus of the cells. These double-strand breaks are all 3 flanked by TET repetitive sequences. This is an artificial integrated construct. In the same cell, we introduce 2 more double-strand breaks, which instead are flanked by LAC sequences. We know that all 5 of them are cut because they are gamma H2X positive. But now in this cell, we transfect an antisense oligonucleotide specific against only the non-coding RNA generated at the TET sites. And as you can see here now, this TET antisense oligonucleotide prevents 53BB1 recruitment to these 3 sites, while within the same nucleus is able to do its job here. So this really proves that with this approach, we can selectively block DDR activation at the individual DNA damage site within the same cells. So yes, we can inhibit DDR in a sequence-specific manner. Is there any good use for this approach? And so this allows me to tell you a little bit of what we have been doing for many years before entering this RNA world, because historically we still are a DNA damage lab that studied telomeres biology and aging, and in particular the role of telomeres in cellular senescence, that is a form of cellular aging. So I remind you what cellular senescence is. You take some normal human fibroblasts, you put them in culture, and eventually they will not be able to further divide because they have reached what we call cellular senescence, or the Hayflick limit, because there is an intrinsic number of cell division that set of cells can achieve. So what impedes the proliferation intrinsically. What impedes intrinsically the proliferation of those cells? Well, what we demonstrated in the past, the telomeres, which are the end of the linear chromosome, cannot be fully replicated at each DNA replication round. The daughter cells inherit telomeres which are shorter and shorter, and eventually telomeres are too short. A too short telomere is perceived by the cell not as a telomere anymore. It is rather perceived as a double-strand break, and like any other double-strand break, will enforce a DNA damage response that will activate a checkpoint that will impede cell cycle progression. So we propose cellular senescence, what we call replicative cellular senescence, as the outcome of the activation of a DNA damage response, which is persistent, protracted, and therefore cells are unable to escape this checkpoint. So this is a good way, probably an appealing way, to explain aging of proliferating cells, but in fact, most cells in our body are non-proliferating. Neurons are certainly non-proliferating, nevertheless, they do age. And it is also known that these neurons accumulate DNA damage. And so we wanted to find an explanation for that. And this explanation comes in the next slide. Again, it's just a scheme to simplify things. But what we did observe that if you generate even random DNA damage, and cells constantly experience DNA damage generation by endogenous sources or exogenous sources, this random DNA damage will generate breaks everywhere, and this will activate DNA damage response everywhere, and this will favor DNA damage repair. However, those lesions that happen to occur within telomeric repeats will not be effectively repaired, and in fact, therefore, you end up accumulating DNA damage within telomeric repeats, also in non-proliferating cells. And therefore, both replicative cellular senescence, the one that I just mentioned, is triggered by telomeric shortening, but also another kind of cellular senescence, which is probably more relevant in non-proliferating cells. They both have something in common, and this is the accumulation of DNA damage within telomeric repeats that we propose, and actually we demonstrated to be irreparable, and therefore causes this persistent DDR activation, this persistent checkpoint activation that we call cellular senescence. And we think this is at the cell level, something which is probably making an impact also at the organismal level. So if we put together the two stories, before I told you that DNA damage triggers transcription that controls DDR, now I'm telling you that telomeres constantly accumulate DNA damage. Of course, it was obvious for us to check whether when a telomere is becoming dysfunctional because it is unprotected, is damaged, is critically short, is unhappy, will this allow the synthesis of non-coding RNA, for the long and the short one? We call them telo-G and telo-C, for the G-rich strand of the telomeres and the C-rich strand of the telomeres. And is this an opportunity to inhibit telomeric DDR with antisense oligonucleotide against the telo-G and telo-C transcripts? So this was the aim, and this is the evidence that indeed when you damage telomeres, you get the induction of this telomeric transcript. So the experimental system that we are using here, we're using a genetic system in which we remove a protein called TRF2 that sits on the telomeres, caps them and protect them. As soon as you remove TRF2, telomeres are not telomeres anymore, are exposed DNA ends, and therefore this triggers a DNA damage response. And again, associated with this telomere uncapping, you get the G strand and the C-rich strand, both for the long RNA and the short RNA. This can then be detected by RT-qPCR, and again, Setu came to help us and indeed could demonstrate that also by single molecule fish you do observe such an induction. Now that we have observed this the ncRNA synthesis, we wanted to check whether we could inhibit DDR specifically at the telomeres with the antisense approach that I just mentioned. And we could demonstrate that both in vitro and in vivo. I want to show you the in vivo data because I think they are more compelling. So by using the same model system that I mentioned before, this genetic inactivation of TRF2, we used an entire mouse in which we could inducibly remove TRF2, so you get a full-blown DNA damage response activation at all telomeres in all the tissues in these mice. Meanwhile, we treated systemically by intraperitoneal injection with a single shot of our antisense oligonucleotide. We collected the tissues and then we tested, we measured DDR activation in these tissues. And as you can see, these are happy mice without DDR. These mice have lost TRF2. You see gamma H2X, you see 53B1, you get this additional marker. These mice also have lost TRF2, you can tell, because gamma H2X is up. However, downstream of gamma H2X, 53B1 is down, phospho-Cap1 is down, and this is the quantification. You really go nearly down to the base level. So this really proves that we can control DNA damage response activation at the telomeres with this approach. And therefore we can uncouple in vivo DNA damage from all its consequences, meaning the DNA damage response and everything which is downstream of the DNA damage response, and which are the many features associated with cellular senescence, and which means, of course, impaired cell proliferation, loss of stemness, of course, the many function of the so-called SASP, which stands for senescence-associated secretory phenotype, which are a bunch of cytokines which are released by senescent cells, which leads to inflammation and fibrosis. Why is this relevant? Why would be good to inhibit DDR at the telomeres? Because there are a number of conditions, not only natural physiological aging, but also a number of pathological conditions which are associated with telomere dysfunction. And this is just a summary of them. In fact, we are together with colleagues at the Mayo Clinic, we are trying to systematizing the field, because there are a number of conditions which either telomeric shortening or telomeric damage at long telomeres that are associated with these conditions. And with our approach, I think we can hopefully demonstrate this is a causal, it's a causal association of telomeric damage and the conditions that, again, I've described you here. Of course, we started with only some, but again, the idea is really to go as broad as possible. So we started with one, which is probably an iconic progeric, meaning accelerated aging condition, is known as Hutchinson-Gilford progeria syndrome, in which many things go wrong, including telomere dysfunction. So we wanted to ask the question, is DDR at the telomeres contributing to this pathological condition? So we started a collaboration with Maria Eriksson at the Karolinska Institute that developed mice in which the progerin, the mutant form of laminate that causes this disease, is specifically expressed in the skin. So we use this skin model of HGPS. We check in the skin whether these mice express this non-coding RNA, which is again are induced upon telomere dysfunction. The answer is yes, but the long and RNA are up. And then what we did, as soon as mice were born, we started to injecting them with this antisense oligonucleotides. Unfortunately, these mice do not live longer than 1 week. That's why we inject them every 3 days. And then we collected the skin and we tested the impact that our treatment had. And this is what we have observed. This is the skin of wild-type mice. This is the skin of HGPS mice. You see this DNA damage response marker, which are up and are to a good extent reduced in mice which have been treated with this antisense oligonucleotide. So yes, we can control DDR and also the consequences of DDR activation, because with this approach, with this TASO stands for telomeric antisense oligonucleotides, we reduce P16, which is a marker, actually is a driver of cellular senescence. We reduce apoptosis and we reduce the inflammation, because we measure the interleukins, like interleukin 8 is down, and therefore this reduction in interleukin 8 also reduces the recruitment of immune cells in the skin of these mice. So there are a number of conditions which are improved. However, we were very curious to see whether we could make an impact on the lifespan of these mice. As I mentioned before, these mice, the phenotype is very, very strong, and this colony of mice never lived longer than 9 days upon our treatment. We extend the maximum survival nearly 50% and 25% mean survival. So to answer the initial question, is telomeric DDR making contribution? Yes. However, we were also aware that this system events take place within weeks, and therefore you may argue that this is distant from what is really happening during physiological aging. Therefore, we accepted the challenge, and so we decided to use a different model. We used the telomerase knockout mice that have telomeres which become shorter and shorter at each breeding. So the third generation of telomerase knockout mice are really, really sick and they really age prematurely. And so what we decided to do was to take the third generation of these telomerase knockout mice. When they are 2 months old, we treat them for 1 month with these systemic injections, but then we let them rest for 9 months. And only when they were 1 year old, we collected the tissues. So here we are going to monitor the long-term impact of our treatments 9 months after the initial, again, IP injection. So now we are collecting, this is very much ongoing, this is unpublished, and we are collecting all the tissues that you have, that you can see here in many different ways. And so far we have been focusing our attention on lungs and the hematopoietic system, that in mice means the spleen and the bone marrow. And what we have observed, so this is the analysis, again, preliminary, still ongoing, in the telomeric antisense oligonucleotide treated mice. So this is the wild type mice. These are the lungs of the telomerase knockout mice treated with a control antisense oligonucleotide. And this is what happens to DDR markers such as 50-DB1 and phospho-CAP1, and this is the quantification in the lungs. So yes, we can control the DDR in the lungs of these mice, and this has an impact beyond DDR. One impact you have already probably appreciated, the structure, the histological composition of these lungs. Indeed, if you just measure the number of air spaces within wild type, the knockout mice, they clearly are, in my opinion, they can hardly breathe. However, because again, breathing means exchanging oxygen on the surface of these cells, of these air spaces. So their numbers dramatically decrease. However, indeed, in the mice treated with this antisense oligonucleotide, to an extent, there is a good rescue. And our pathology is digging deeper and deeper and is scoring many different parameters. And overall, the pathological score, that means the higher it is this bar, the sicker is the mice, the sicker is the more pathological is the tissue. Again, there is certainly a very striking difference between the wild type and the knockouts. And again, to a good extent, this treatment reduces this pathological score. And now we are performing RNA-seq experiments, and the data are extremely exciting, extremely encouraging. We see a very strong rescue of many of the pathways which are usually associated with aging. Here I'm just spotting the mTOR signaling, inflammation, P53, IL-6, JAK-STAT signaling. So this is again preliminary, and we want to go much deeper. But suffice to say that basically what this really seems that these antisense oligonucleotides are effective tools, to the least, to test the impact of telomeric shortening in the lungs. So they can really, they are really a tool to answer the question, is telomeric DDR contributing to this condition? And the other thing that we, again, I'm not showing all the data, but certainly we could demonstrate that these oligos reach the lungs, they have long-term effects, they are very effective in inhibiting DDR. They have an impact on the histological structure of these mice, and they reduce inflammation. And we also have very strong evidence in the reduction of fibrosis. And the pathways, and we are just scratching the surface, the many pathways that are impacted. So we are now extending this observation not only to the lung, but also to the hematopoietic system. Spleen is a hematopoietic organ. In mice, again, here, luckily, we have a very good pathologist that help us in determining the impact that this that the inactivation of telomerase has on these mice. This is the so-called white pulp in the spleen of this wild type mice, which is seriously compromised in the telomerase knockout mice treated with a control agent, but in the anti-telogene, anti-telocy treatment, this is significantly improved. And again, the pathological score, which is up in the knockout mice, is reined in, is controlled by these treatments. And there are, again, many more pathways that now we are many other features that we are characterizing. And in addition, the bone marrow, which is again an additional hematopoietic organ, again is compromised. And again, here we are performing additional experiment to again go deeper into the impact that telomerase inactivation has and the rescue of our antisense oligonucleotides. So again, also in these organs, we see an impact, and overall the biological impact is that the myeloid skewing, which is a typical feature of physiological aging, which is exacerbated in this telomerase knockout mice, is again to a good extent controlled by our treatment. So what I told you basically is that we have a novel tool with, I believe, also with some therapeutic potential these are the telomeric antisense oligonucleotide, they inhibit DDR at damaged telomeres and they are affected in vivo, and they restore tissue homeostasis. And I showed you skin in the HGPS mice, I showed you lung, I showed you bone marrow, spleen. We have evidence also in other tissues and also in other models where these antisense oligonucleotides can extend the lifespan in independent animal models. Again, HGPS telomerase did not have the time today to show you, but we have also a completely independent anemic mouse model in which we have a dramatic rescue of the lifespan and of course of anemia of these mice, again anemia triggered by telomere dysfunction. And we observed this in different species. I showed you mice just because of lack of time, but we have some interesting results also in zebrafish. And again, this is independent from the genetic cause. So progeria is caused by lamin mutation, telomerase is caused by telomerase inactivation. So provided that a condition causes telomere dysfunction, this is an approach that could be useful. And so that's why we are excited, but most importantly, why are we excited? Because here, what we are really doing, we are seeing a lifespan extension by targeting ncRNA, which is neither coding nor actually encoded by any gene. So that's why I'm quite excited about it. So I'm finishing here. I cannot not take this opportunity to mention that we are always looking for talented postdocs, especially if they are experienced in RNA biology, and as you can see, in mouse aging studies. And of course, we are always willing to collaborate with anybody who's willing to use these as tools to determine the impact of telomere dysfunction in their system. This is us. I think I mentioned, or at least I've shown the name of the authors of the published results. Here in red, rather, I've highlighted the people that have generated the last set of results on the telomerase knockout mice, which are still unpublished. Claudio Tripodi is our pathologist. Valeria is in charge of the immunohistochemical analysis. Sarah Sepp is our mouse person. Francesca is our telomere person. And Fabio is our computational scientist that has been taking care of the RNA-seq data. So I'll stop here, and I'll be happy to take questions.
Oh, great. Thank you very much, Fabrizio. That was amazing. Any questions from the audience? Please unmute yourselves and ask your questions, or feel free to type them into the chat and I will ask the question. So first question comes from Dan Tenen. He says, maybe I missed it, but what is the molecular basis of how the non-coding RNAs recruit the POL2 complex at sites of DNA damage? Do RNAs recruit the POL2 complex to regular coding genes as well? And is it the same mechanism?
So the RNA POL2 is not recruited by the RNAs. It's the mRNA complex that recruits RNA POL2. So the sequence of events, is the way we see it, is you get a double-strand break. This is sensed by the mRNA complex. mRNA binds to the double-strand breaks, then recruits at the sites of DNA damage RNA POL2, and the RNA POL2 will start transcribing this non-coding RNA. Yes, these events take place also at both coding and non-coding regions. Yes.
Judy, go ahead.
Yeah, I had a question that was very interesting. First, I want to know, does the length of the PAL2 transcript have anything to do with the size of the foci? You said that it was really downstream of gamma H2AX. So does gamma H2AX determine the size of the foci, but does that then determine the length of the RNA?
That is unlikely to be because, again, gamma H2X spread for hundreds of kilobases, so I cannot imagine transcript that long. So these seem to be two different events. And we think that the RNA is somehow seeding contributing to this positive feedback loop that I mentioned before.
And then another question was, isn't it bad for the cell to have unrepaired double strand breaks? And why isn't that activating P53 and downstream?
Okay. It would be bad if you leave a double strand break in the middle of a chromosome unrepaired. That is bad. But if you have just a signaling chromosome end, which keeps the checkpoint enforced, impede that cell to proliferate and start transcribing inflammatory cytokines, if you relax that checkpoint, you will allow that cell with just a bruised chromosome end to proliferate, but that will not lead to genome instability. I should mention that in these mice, we did not see any evidence of cancer. And what I failed to mention is that with these antisense oligos, we do not only blunt the signaling, but also we impede the DNA repair. Therefore, we prevent chromosomal fusions, which of course could be a source of genome instability.
But when the cells replicate their DNA, doesn't the double strand break?
No, because it's a very tip of the chromosome. So basically, you would have just the full chromosome fully replicated without any consequences.
You wouldn't have shortened telomeres?
No, because again, so it certainly, yeah, you would, if the double strand break is within the telomeric repeats, you may lose the very last terminal bit. Yeah.
Thank you.
So I have a question. So you should be applauded for trying to pick a very aggressive mouse model to try and rescue with your experiments, or a couple of very aggressive models. I wonder if you've thought about something with a slight loss of function and telomere function, something like some of the dyskeratosis models as a starting point, which doesn't mean that you've got a complete loss of the telomeres or a complete loss of telomerase. Does this make sense to you?
Yeah, no, absolutely. We could go even in the direction that you are pointing and go even further and attempt to treat wild-type mice, which in any way, physiologically, they will accumulate DNA damage at their telomeres. And then we should be able to see, I really would be interested in seeing whether with this approach, we can extend the lifespan also of wild-type animals. But yeah, you're right.
I see that as being not such a fun experiment if you're going to inject them twice weekly for 2 years.
No, because I showed you that the telomerase knockouts, we inject them for the first month, and then we let them uninjected for 1 year, and then we still see an effect. So possibly we may have long-term effects.
Sure. Okay.
Hi, hello, just a quick question. Thank you for the talk, very interesting. Do you have any idea of what's the role of the short RNA that you describe in your talk, and they're able to talk to the long non-coding, and why they are important if they are recruiting other proteins, why they don't lead to any microRNA-like degradation?
Well, we cannot exclude that when ddRNA binds to the long delinker, and this does not lead to its processing. This is something that we have fantasized about, but we never demonstrated. I suspect that this is a sort of a feedback loop, and so what we really would like to do, and this is what we are doing in the lab, we are reconstituting these events in vitro, and I'm really keen to test whether the long RNA and the short RNA have different roles in modulating the viscosity. So there is of these forces, there is already evidence that equal amounts of equimolar amounts of the same RNA, but with different sizes, with different lengths, have a different impact on the viscosity of different LLPS events. So I suspect that the short and the long RNA, but again, certainly we did not demonstrate that, we don't have any evidence for that, but just in my mind, it would be interesting to test whether long and short RNA have different functions in modulating the viscosity of these condensates.
Thank you.
I have a question for you, Fabrizio. Great talk as always. So do you take telomeric antisense yourself?
No, not yet, not yet. I'm waiting for FDA to approve that.
Okay, okay. So another question, I might have asked you this before. It's with Cas9 induced double-strand breaks. It's thought that the Cas9 complex sits on one side of the DNA. Do you see just a one-sided RNA production and not the other direction?
Excellent question. We do not know the answer. So meaning that we want to have, in fact, probably the best experiment would be to have the Cas9 cutting the same site on the two strands, sitting on one strand or the other strand, and then monitor at the same site the impact that this has on the link RNA production. We never did. We have not done that experiment yet, but yeah, it would be interesting.
Okay.
And I agree with you that CRISPR sitting there is certainly bothering to an extent the DDR machinery.
Right, and it's known, it gives you gamma H2X just fine, but repair is very, sluggish, and the rate of death is pretty high for that, single double strand break. So I'm wondering if you're sort of messing with the production of the D-link RNA, and you can't really assemble what you need to do to repair it afterwards.
That's quite possible. We don't have a definite answer. No.
Yeah, great talk. Thanks.
Thank you, Matt.
So we have a question apropos Matt's question from Sophia Adamia. She asks, could you talk a little bit about the toxicity of your TASOs? Is there any systemic toxicity?
No, so we did some MTD experiment, meaning some maximum tolerated dose experiments. We can go pretty high. We have not seen long-term consequences. Of course, our experiments are limited to the time length of the experiment that I showed you, but so far, no.
A question from Hungo Lee. Here she asks, that's a very interesting, great talk. Repressive histone marks like H2AK119UB and H3 K27 methyl 3 are known to be recruited to double-stranded break sites as well. Do you know how long the transcription lasts after double-stranded break, and would the repressive histone marks be affected by ddRNAs?
Yeah, beautiful question. We do not know, and you know why? Because we don't have a tool to answer this crucial question, meaning we don't have a way to introduce in a site-specific manner a double-strand break and then switch off the cutting enzyme, because CRISPR will keep cutting. And so what I showed you is the result of cutting. It's not a hit, because X-ray would do hit and a single hit, but again, would not be site-specific. So we don't have a tool to introduce a double-strand break once and only once in a cell, unfortunately.
All right, very cool. Question from Tribhuwan Nadav, who asked, why are the DNA damage transcripts generated breaks a few kilobases, and what stops their elongation?
Yeah, so we are presently characterizing the determinator complexes, or whatever, which are the proteins which control the termination. Yeah, this is in progress.
Okay, good. And then from Richard, is mRNA's recruitment of POL2 to the foci site direct, or is it a non-specific effect due to the generation of single-stranded DNA at the break site?
I didn't get it. So is mRNA, say it again?
Is the recruitment of POL2 by mRNA at the foci site direct?
Yeah, well, certainly, okay, there is a protein-protein interaction. So if you do an IP of mRNA, you will bring down RNA pool 2, but I can tell you more. This interaction is much stronger if you do such an IP from cells which have been previously irradiated. So I suspect there are some post-translational modifications that control such interaction. Is it direct? I do not know in vivo, in a cell. However, the fact that I can reconstitute these events by just mixing purified recombinant mRNA and native purified RNA pool 2 makes me believe that the two proteins can at least functionally interact directly.
All right, fantastic. So are there any other questions? Feel free to unmute yourself. All right, if not, well, let's thank our seminar speaker again. That was really a very thought-provoking seminar, and I hopefully will open up lots of collaborations with individuals who are on the seminar. So thanks once again, and we'll see everybody next week for our next event.
Thank you. Thanks, everybody. Thank you.
Bye.